Histological techniques are the different methods used for preparation of microscopical sections. Sectional method involves cutting the tissues into very thin slices or sections for light microscope and ultra-thin sections for electron microscope. In this method, the relation of the cells to one another is preserved, not disturbed. Spreading technique and smears. Spreading technique is used to spread tissue sheets on slide, like loose areolar connective tissue, while smears on glass slides are used for studying suspensions like blood, urine, and bone marrow. Tissue culture is a new method in which the tissues are cultured in a special media in order to study their characters and properties. Microtechniques are the different methods for preparation of microscopical sections. For light microscopy, there are three methods of sectioning: the Paraffin method, the most common, the celloidine method, the most ideal, and the freezing method, the most rapid. Paraffin method. Preparation of paraffin sections. These are the various stages through which histological sections can be prepared. Fixation. A small piece of the organ is obtained from the body immediately after death and placed in the proper fixative. Many fixatives could be used, as formol saline, Sousa, Zinker, and Buen. The type of the used fixative depends on the tissues and the stain. For example, soft tissues need a special fixative than hard one. Also, the different stains. Fixation prevents putrefaction and autolysis of the tissues by coagulation of their proteins. Also, fixation hardens the tissue, facilitates cutting, and increases its affinity to different stains. Washing is done by running tap water to remove the excess of the fixative. Dehydration. Water is removed from the tissues by placing them in ascending grades of alcohol, 70%, 90%, and 100, in order to remove water gradually from the tissues to avoid their shrinkage. Clearing is done by placing the tissues into a paraffin solvent, as alcohol used in dehydration is not miscible with the wax in which the tissues are finally going to be embedded. The paraffin solvent used is xylol or benzol. By placing the tissues into the paraffin solvent, the tissues become translucent and clear. So, this step is called clearing. Impregnation, in which there is replacement of the paraffin solvent, xylol or benzol, with a molten paraffin wax. This is done in the oven. First, the tissues are placed in soft paraffin, having melting point 50 degrees centigrade, then in hard paraffin with a melting point 58 degrees centigrade. This hardens the tissue. The time for impregnation depends on the size of the tissue. Embedding. This is done by using a special hollowed metal box placed on a movable glass plate. Molten hard paraffin is poured in the metal box smeared with glycerine, and then the tissue is oriented in the center. On cooling, a paraffin block is obtained with the tissue in the center. The movable glass plate is removed and the block can be easily taken. Embedding is important as it supports the tissue and facilitates cutting. Cutting. The paraffin block is attached to the holder of the microtome, and sections are cut. The thickness of the sections ranges from 7 to 10 micron. Mounting. This is done first by placing the paraffin sections in water bath having 37 degrees centigrade in order to spread the sections. Then the sections are mounted on clean glass slides wrapped with sticky material like egg albumin. Staining. Sections are now ready to be stained. The common stain used is hematoxylin and eosin. Hematoxylin is a basic stain with a blue color, while eosin is an acidic stain with a red color. Structures that accept the blue color of hematoxylin are called basophilic. They are acidic in nature. The reverse in the case of structures that accept the red color of eosin. They are called acidophilic. They are basic in nature. Nuclei are always basophilic because of their content of nucleic acid. Cytoplasm may be acidophilic or basophilic. Basophilic cytoplasm indicates activity due to the presence of nucleic acids. Steps for hematoxylin and eosin staining. The idea is that sections on the slides are impregnated and surrounded with paraffin wax, and most of the stains used are dissolved in water. Wax is not miscible with water, so the tissues cannot accept the stain, so wax has to be removed by xylol. Again, xylol is not miscible with water, but is miscible with absolute alcohol. So, xylol has to be removed with absolute alcohol, then gradually replace the alcohol with water. This process is called hydration. Is done by placing the sections in 90%, then 70% alcohol, then in water. Remove paraffin from sections by placing them in 2 changes of xylol, 2 minutes each. Hydration is done by placing the sections in descending grades of alcohol, absolute, then 90, then 70. Put the sections in distilled water. Stain in hematoxylin for nearly 1 minute. Then put the sections in tap water for bluing. Rinse the sections in distilled water. Stain in eosin for nearly half a minute. Wash in distilled water. Cover the sections by putting a drop of glycerine jelly on the slides, then cover with a cover glass. Glycerin jelly is miscible with water, but if you are going to use a sticky material like Canada balsam, it is not miscible with water, but only with xylol. So you have to return to the same steps in the reverse manner, that is to say, removing the water by gradual dehydration in ascending grades of alcohol, 70, then 90, then absolute, 2 changes. Then put the sections in xylol for clearing, put a drop of Canada balsam and apply a cover. Siloidine method. Steps: fixation, washing and dehydration are carried as usual as in the paraffin method. Clearing, however, is done in a mixture of equal volumes of ether and absolute alcohol, which is the siloidine solvent. Impregnation is carried at room temperature first in thin celloidine 7.5%, then in thick celloidine 15% for long time, that ranges from 15 to 30 days in each. Celloidine blocks are prepared and preserved in 70% alcohol until sections are cut and stained as usual. Freezing method. Steps. Specimens are frozen in the cryostat, and frozen blocks containing the specimens in the center are prepared and attached to the holder of the cryostat by tissue tape. Sections are cut and stained as usual. Comparison between the different methods for light microscope. The advantages of one method are disadvantages for the others. For example, advantages of the Paraffin sections are short period of time, gives very thin and serial sections. Sections are easily cut and stained. Disadvantages of the celloidine method: long period of time, gives very thick and no serial sections. Sections are difficult to cut, mount, and stain. The disadvantages of freezing method: gives thick and no serial sections. Sections are not easily cut, mounted on the slide or stained, but it has short period of time. Disadvantages of the Paraffin method are damage of the tissue by overheating. Lipids and enzymes are not preserved. These disadvantages are advantages in the other methods. Advantages of celloidine method: gives perfect sections as no heat is used. It is excellent for large pieces of tissue. It is better for soft tissues as brain. Advantages of freezing methods are: it is the most rapid method. It is excellent for histochemistry. It is used to diagnose tumors during surgical operations. Some special methods of staining. Vital staining is the staining of a living cell inside the body by using a vital stain, as neutral red or trypan blue. It is used to stain phagocytic cells. Supravital staining is the staining of a living tissue outside the body, as a staining of the blood with brilliant chrysal blue to detect immature blood cells like reticulocytes. Metachromatic staining is the staining of a tissue by a color that differs from the color of the original stain, as the granules of the mast cells, which accept a red color after staining with toluidine blue. Silver staining, using silver nitrate for demonstration of reticular and nerve fibers. Silver impregnation, that is, impregnation of the tissue immediately after death in silver nitrate. It is used to demonstrate Golgi complex as in nerve cells and epididymis. Histochemical methods used for identification of either inorganic substances as iron and calcium or organic substances as enzymes, for example, acid alkaline phosphatases. Immunohistochemistry uses antibodies to specific cell molecules to detect their presence in the tissue sections. Fluorescent and immunofluorescent methods, in which certain substances and antibodies are labeled with a fluorescent dye to emit visible light to be detected by the fluorescent microscope. Important measures used in histology. Micrometer equals 1/1000 of the millimeter. Nanometer equals 1/1000 of the micron. Angstrom equals 1/10,000 of the micron. Preparation of sections for electron microscope. Steps. Fixation. Very small pieces are obtained and immediately put in glutaraldehyde followed by osmium. This is the fixative for electron microscope. Washing is done in phosphate buffer 3 times 10 minutes each, then in 3 changes of distilled water 2 minutes each. Accurate dehydration is carried out in ascending grades of alcohol: 30%, 50%, 70%, 90%, and then 100% each for 15 minutes. Clearing is carried out through a mixture of propylene oxide and araldite. Impregnation is done by using 2 changes of araldite. Embedding is performed in plastic media. Cutting. Ultra-thin sections are cut by the ultramicrotome using diamond or glass knives. Mounting. Sections are mounted on copper grids and stained with uranyl acetate and lead citrate.